Because you guys are looking at me like, which world, which planet this guy came from? He's speaking a language that I don't understand. My MSc biotechnology or bachelor's of biology guys. It's unfortunate, but come on, speak up. You can speak about, right? Really, go do this exercise to your parents. It's going to be fun. No harm, right? You say something to your parents all the time. You say, oh, you didn't raise me well, right? You could have done this better and that better. How many of you have said that to your parents? Don't tell me. Don't lie. I'm sure most of you have said that, oh, you could have done this better when I was growing up. No? Ah, you guys are all thankful to your parents. How cool. Very nice people out here. That's unlike me. I was horrible. I told my father all the time, I wanted to be a doctor. You made me an engineer. I did this all the time. I'm sure you guys do this. Something like that. No? Okay. All right. That's fine. The point is very simple. Can you explain this to somebody who does not understand what RNA means? Go home and try and explain to your father first, what does DNA mean? I mean, come on, he should know. He has lots of DNA, right? He should know how he was built. What kind of animal your father or mother is. How bad does this sound? Because we're all animals at the end of the day, right? We evolved from monkeys, guys. And monkeys evolved from something, that evolved from something, that evolved from something, and that evolved from something. And we all evolved from carbon and hydrogens, right? The Mother Nature. I am sure your parents believe in Mother Nature. And you can explain that to them, how that works. Okay, just do that for me, okay? All right, good. We talked about this figure. I'm sure you've seen this figure 10 times by now. People who are attending the classes, right? There's a histone, here's a nucleosome, there's a DNA wrapped around it, there is tail, unstructured tail, there is lots of different kinds of modifications that happen, which are called histone modifications. We've been talking the same thing for the last 2 days. Who has doubts? I put this slide only for you. Who has doubts? Yes. Go on. I really want to hear doubts for next 10 minutes.
Syndrome, and it showed that there was epigenetic, like not just the trisomy of the 21st chromosome, but they also suggested that there was hypermethylation or even hypomethylation in the gene in the 21st chromosome. So can we, like, is it possible that we could correct it somehow, or we could use repressors of methylation to somehow suppress the effects of Down syndrome?
Can you use epigenetic modifiers to change trisomy 21? That's what you're asking. Can I change the status of this or reverse it? Yeah, you can. Nobody's done it. Go on, do it. Right? In theory, that's possible, but nobody's done it. I'm telling you. You can try it. Okay. A 10th grader from New York came to my lab once and said, I have a project in mind. I want to do it in your lab. And I'm not kidding, this is a real story, okay? So this guy wrote to me in an email and said, Kunal, I have an idea for you. He said, I'll be Kunal, not sir, okay? I said, okay, please. And he said, there are oncogenes. Do you guys understand oncogenes? Okay, MYC, right? We talked about MYC, an oncogene that causes cancer. Can I use an epigenetic repressor to silence an oncogene? Just like what he's asking. I was like, very good, 10th grader, come to my lab and try the experiment. He came to my lab all the way from New York. His father came with him. He spent 3 months, three months in my lab. He came back again in the winter and he spent like 15 days. He had holidays between 10th and 11th grade. He came to my lab and he did it. He did a better job than my postdocs and PhDs. Many of you are looking blankly at me at what epigenetic repressor means. Okay? He went on to study at Caltech. As you guys understand Caltech, he got admission from Stanford, Harvard, MIT. And my recommendation letter to him said, I have never seen as smart a guy as him, and he was better than my PhD students. I was very proud of writing that, but at the same time, I was very sad at writing that. Right? The guy studies PhD in MIT right now. Okay? He's only 23 or 24 years old. If somebody like that, 10th grader, can come to my lab and do this, I'm sure you guys can, right? He has a good idea. He can try that, right? Okay. It's very challenging. It's not possible probably, but what's a life lived without taking the impossible challenges, right? All these big people say things to you guys and you hear them out and you say, okay, I'm not going to do it. I'm going to go home and watch a YouTube video. Okay, I have nothing against YouTube videos, as I told earlier. I'm just propagating my Mr. Sundar Pichai's company, okay? All right, how you can see in this slide earlier. Did you have anybody has doubts? Okay, well, you and I will talk later, okay? I want everybody else to, sorry. I know you're very smart, you're thinking a lot, very good. Rest of you. Has anybody have doubts? Confusion? Yes.
How do this H3K79 methylation control the number of transcripts?
How does K79 methylation controls transcription?
Number of transcript form. You said like it control how many number of transcript it form. It is based on this mark.
It is a memory of transcription. It is a certain level of it starts to inhibit how RNA polymerase is going to go forward or not. So it's an auto feedback loop at some point. That's a more complicated question. Let's talk later. Okay. I want you to. I mean, I know you are also you're from epigenetics field or something. No, no, you've studied, which is good. You came from somewhere else, right? You came from Kolkata. Good. Okay, so he's asking a more complicated question that I don't want the rest of the class to feel sad about. So I'm just going to talk to you later. Okay. All right. Anyone else has simple doubts about histone modifications? Because I'm going to move on to do other things. Okay? No? Everybody understands histone modifications? Great. Histone modifications have specific patterns, right? I'm actually not going to move on because somebody gave me a feedback yesterday that I should talk about how we use these things in the research. So I'm going to tell you a story from my lab. Okay? Very simplistic. So the questions that we ask in my lab is how does epigenetic landscape gets altered in cancer? How does the chromatin states, the picture I was showing you earlier, get altered in cancer? So I'm going to show you a very simple story, and I expect half of you to not get it, but I expect those half of you to stand up and ask me question. That wouldn't be nice? Would that be nice? Because again, you guys are BScs or MScs and you can ask questions, right? And you should get part of it. So, and there will be a cancer epigenetics class down the line. Somebody will explain this too. They won't explain my research, of course. So I'm just going to do two minutes of justice to application of what we are hearing, because not just the theory, okay? The application is okay. So how do the chromatin states change when you go from normal to tumor? That's all I want you to know. Look in this slide, okay? There's a blue arrow that says normal, tumor, chromatin states, right? Only this portion. You guys get this? That's what I want you to focus on. That's what we do. So we made some models of tumor initiation in a cancer called melanoma. This is a cancer of Western world, not Indians for the most part. Okay? It happens a lot in Australia. It happens a lot in Europe and America. So they spend a lot of money solving melanoma. But it does happen in India too. In fact, I know a friend of mine who got melanoma in Bilaspur. Okay? It's a disease of melanocytes. You guys know melanocytes? Okay, melanocytes are what make you brown or black or white or fair and lovely, right? Okay? But these days there is something else, right? They call it something else. The people take offense to this. All right. More melanocytes, you are darker. Less melanocytes, you are everybody knows this. This is very well put in the system. Okay, white melanocytes, when you have less melanin, that's actually works against you. So be proud of you being brown, okay? Because that prevents melanoma. So that's why melanoma is not found a lot in. Somebody could come back and challenge me. But Kunal, there is lots of black people in America and they get melanoma. How is that? You just told me something wrong, which I did actually. But there is certain melanin synthesis pathway in the brown people. Somehow it protects us. okay, for whatever reason. Or we might have something else in our genetics, and that's okay. And I don't know, nobody knows the answer to this question. Great question for you to do your PhD on. Why do Indians not get melanoma? Can you make Indians get melanoma? Hey, that would be fun. No? We'll have one more thing the pharma company can make money off, right? Okay, so we make model systems for this. We took melanocytes, lightly pigmented melanocytes, because white people have them, right? We induced some genetic changes in that, and we said, boom, when I induce this change called P10 loss, P10 is a specific protein in the cell, so don't worry about P10. It's a very important tumor suppressor. It happens in about 20% of all cancers of P10 loss, okay? P10, we did P10 loss, and now you see this curve going down. How many of you know what kind of curve this is called? None of you, I'm pretty sure. It's called Kaplan-Meier curve, okay? It is plotting survival. So as you go down, that means the person was dead, okay? The mouse in this case was dead. But this is used all the time in clinic to plot survival of any kind. Doesn't matter, cancer, can be diabetes, can be anything else, okay? Can be any kinds. This is, you're dead, you're dead, you're dead, you're dead, you're dead, you're dead. Most of the mice are dead here. Okay, that means these people were dead, these people were not. They were surviving, so the line is flat. That means those mice were good, happy. Okay, so when I inject these cell lines in the mice, these mice are fine, these mice are not. Okay, that means, and they had tumors. Okay, they had tumors in their skin coming from their melanocytes, because we injected the melanocytes, of course, right? So that's called Kaplan-Meier curve. These cells are tumorigenic and these cells are not. Okay, that's all. So when I lost P10, those cells make tumors. No surprises, tumor suppressor genes. When you lose tumor suppressor genes, they make tumor. How many of you got that sentence? Okay, thank you. Are you guys listening, by the way, on the back? Really? So you understand English, right? So when I say, did you get the sentence? Do you get the sentence? Or am I speaking some other language? So then say yes. Raise your hand. Raise your hand. ATP burn, calorie burn, it's going to make you thinner. Okay? Good for you. I'm surprised. Answer, guys. Respond. Your teacher is asking you something. Respond. Okay? Is that clear? If your wife or husband at some point in your life asks you something, you're going to have to respond. Otherwise, you'll have a fight. Okay? I'm making silly jokes all the time here, right? Actually, I should watch my own YouTube videos and give the collection of these silly jokes I put. I actually am not that funny. Okay, so I taught you guys ChIP-seq yesterday. How many of you got ChIP-seq? Right? Chromatin immunoprecipitation. For rest of you guys, there's a chromatin, there is an antibody, and you precipitate it. Okay? English, chromatin, immuno means antibody here in this case, precipitation means you pull down. So I took my cell extract, I pulled down my antibody, with my antibody, that point of interest, I got that DNA, and I subjected it to the next generation sequencing I taught you earlier, okay? So we got what? We got regions where specific antibody bound regions were there, okay? We're not going to worry about the details of it, but that's good enough for you for now. Okay, so in my lab, we developed something called high throughput chip sequencing, meaning we can do lots of histone modification marks in lots of different systems. Because yesterday I just told you there are more than 150 histone modifications in the cell, and we want to understand the combinatorial patterns of those histone modification marks. How many of you got what I just said? Okay, I speak fast and I speak English, but I can speak the same thing in Hindi. Did you guys get it? No. No. Okay. I think many of you don't get it. So let me go back to this picture. This picture shows you a nucleosome. A nucleosome has multiple modifications, but not all modifications happen in the same nucleosome. There is a specific combination of modifications that happen on a given nucleosome. Okay? So that specific combination, if you heard combination permutation, all descendants of Aryabhats, right? You understand math, right? Combination, permutation, combination. Combination means specific sets of histone modifications occur on specific tails. That's called chromatin state. Okay? So we pull them down. We pulled out each of, sorry, to determine the chromatin states for these 40 or 37 different histone modification marks, we pull them down in those cells which are called tumorigenic cells and non-tumorigenic cells. What are we trying to ask? We are asking what kind of chromatin modification states change as we become tumor from a non-tumor. Very simple. Okay? A cell is non-tumor and a cell is tumor. Okay, and I'm just asking which chromatin state changed. Okay, so we generated a lot of ChIP-seq data using high throughput chip sequencing, and we got something like this. This is real world data. Okay, what do you see on the screen? A very complicated pink picture, right? Oh, there's no pink here. I'm sorry. This was all blue, and on my screen it's pink. Okay, basically, these are tracks of these histone modifications in a non-tumor and a tumor, non-tumor, tumor, non-tumor, tumor. You see blue, red, blue, red, blue, red, blue, red, right? Those are peaks. For those of you who didn't come yesterday, those are called peaks. When you do chromatin immunoprecipitation or ChIP, you get enrichment of those DNA fragments that we talked about on given locations in the genome. Okay? And that makes a peak like this. So those pileups are peaks. Okay? So you see this is K27 trimethylation here. You see that? K27 trimethylation is here. This is a silent region. This whole region is silent. There is no other activity. And of course, you can see that because there is no other signal in any other else except this place, right? So when there is a K27 trimethyl mark, there is no other signal, which is a polycomb repressive mark. So of course, when there is a repression mark, active mark cannot be there, right? Cannot be, right? So when you're silent, you're silent. So rest of the active marks are not there. Here, a detailed view of a gene here. You see a gene on top? Okay. So you get these kind of signals of multiple kinds and active marks because on the bottom, everything is acetylation. And what did I tell you? Acetylation equals to gene activation. Beautiful data, right? I love the data from my lab. Of course, I do because it's in my lab, but it also costs a lot of millions of dollars. So I do. So you see lots of blues, but you'd see very little red, right? Blue is non-tumor. Red is? Okay. So blue means, and loss of peaks in the red signal. That means I don't have any more of acetylation or I have little acetylation remaining. Okay. So I found out that when I make a non-tumorigenic melanocyte into a tumorigenic melanocyte, I lose acetylations because all these things on the bottom, you see, acetyl, 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 acetyl. Did you guys see that? Okay, there are different kinds of acetylation. And I told you guys also, don't worry about acetylations, just remember one acetylation. We did not know that at that point. We thought they may have different patterns. But what we found out was, after spending a lot of money, that all acetylations don't care. They're actually there in most places. I just lied. Actually, we did figure out more acetylations were different, but for you guys, don't have to worry about it, okay? Most acetylations are positive, okay? We also did RNA-seq on these. How cool. We just learned RNA-seq, right? Didn't we? Okay, 5 minutes. So you see here, there is a change in expression. Is there a change in expression? You see any? There is change in expression. There was more blue, there's less red. You see this peak for particularly? You can see that, okay? So we lost acetylation, we lost the gene expression. Of course, that was meant to happen, right? Histone modifications control this. Okay, but there are some other marks that don't change. You see, these marks don't change. These marks don't change. These marks don't change. Specific chromatin states are changed, and that results in gene expression problems in a non-tumorigenic to a tumorigenic behavior. Okay, in melanoma, doesn't matter. You just care about melanoma cancer, cancer. I got something. Okay, okay. We then built chromatin state models for these. Okay, what was chromatin state? A nucleosome on the bottom on the right side has specific sets of marks because my previous slide just told me that at a given location, so if I just look at this location, I have a lot of acetylation. At a different location, I have some of them. Okay, at a different location, I don't have any of them. So if I segment my genome into, let's say, 100 1,000 base pairs, and I said, okay, my segment was a 3.3 billion bases genome. I segment them like this, okay? Just go and segment them, and I count how many peaks are here, how many peaks are here, how many peaks are here. I would be able to identify what peaks are enriched, right? And I'll be able to say, in this nucleosome that is present in that 1,000 base pair, the X chromatin state is available. Did I get it? I think most of you didn't get it, or most of you got it. You got it, right? A combination, very simple. You see, these peaks are at a given place, right? But this place doesn't have many of these marks here. Okay, so we've called chromatin state, and that's what is shown here. This is region 1. You see region 1 has mark A, B, no C, D, and E. Okay, mark region 2 has 1, 2, 3, 4, 5, all 5 of them. Whoa, nice. Region 4 has 1, 2, no 1, and then some of that, right? So on and so forth. Region 5 has 1, 2, and 3, but not 4 and 5. That's what is called chromatin states, combinatorial patterns. Okay, so we looked at combinatorial patterns, and that's expressed here on the bottom with simple blue and white heat map. Now I didn't use red because some of the colorblind people cannot read red. Okay, so you make white to blue. So you care about your audience, right? Anyways, so you see the patterns are visible, right? That came out of a machine learning model. Okay, I'm teaching you the most cutting edge epigenomics you've ever seen in your life for people who don't understand PCR. Okay, so we did an 18-state chromatin model. That's a model of chromatin states. So that many chromatin states exist in these cells. Okay, which is cool because now for the first time ever, I've defined chromatin states in melanomas. That's why our paper got published. Okay, all right, but it's not about paper publishing, right? Is it? It's not about paper publishing, it's about curing people, right? So we got this chromatin state models where you see blue combinations, then you see some blues, some others, right? You see different combinations. This is a combinatorial patterns that are different from each other. There are 18 different chromatin combinatorial patterns. Again, my descendants of Aryabhat, you guys need to think about combinatorial patterns. It's a complicated problem. Not everybody gets it, and that's why I don't expect you to get it. So if you don't, then don't. But if you do, great. On the bottom one most, I get blue everywhere. Do you see that? So that's my artifact state because it's an experiment. I told you yesterday, your experiment is as good as your control. This is a control because my control was IgG. You see that IgG that was enriched there. So I said, boom, I'm not going to care about that state because I know that's just artifact of my ChIP-seq experiment. Okay? Got it? But rest of it are there because IgG has no clue. Boom, I got it. I like it. I have confidence in my data. As an experimentalist, I need to have confidence in my data to be able to interpret the data. That's what I was telling you yesterday. Okay? Got that?
Yes, sir.
Okay, so there are different chromatin states, I have different things. I'm not bothered about it. There are promoters, enhancers, transcribed enhancers on the left side. Can you guys read that? Transcribed states are K79 methylation. The question he just asked me, right? And then low states have no marking, meaning most of the nucleosomes don't have any marking, or my experiment is not good enough to be able to define that because it's not sensitive enough. It's possible, okay? I have polycomb, which is K27 trimethylation. Polycomb, when it's there, nothing else is, no active marks are there, right? We saw that on the screen as well, right? So when I have polycomb, when I'm suppressed, I'm not active. Of course, obviously, heterochromatin. I have no other thing other than heterochromatin. You see that? And I told you yesterday, if you have K9 trimethyl, you don't have K27 trimethyl. There are different ways of gene repression, okay? You get it? That's why I say that. Because I have data to support my call. I have data to support my conclusion, okay? And this is heterochromatin. That's at repeat elements. It's killing long-term survival. It's killing for long-term. Quiescent state is something where there's no IgG, right? And there's no other signal. So I say, okay, this is quiescent, meaning there's probably not much going on, okay? Anyways, we can talk all about it, but bottom line is you got the combinatorial patterns of chromatin state in a real-world data. That's a real-world data, and we can keep going there. And then, importantly, we modified distances, highly acetylated, less acetylated. The reason for that was very simple. I'll show you that in the next slide. When you look at the differences between non-tumorigenic states to tumorigenic states in this kind of model, which I'm sure most of you are confused about right now, right? Why would you be confused? What we are looking at a transition of the same state between two different cells. So I'm saying, okay, my state number 1, the combinatorial pattern 1, how did that transition between a non-tumor to a tumor? And I said, when it goes off diagonal, so diagonal will be obviously always there. Why will it always be there? Because most of them will be a steady state, okay, will be there. But when I see off diagonal changes in blue, that means that transition. So my state 1 transition to my state 2 and 3. Okay, that's where my most blue is, right? So that's the most amount of epigenetic differences that occurred between a non-tumor and a tumor cell. Okay, cool? Very nice. So I can, and then I looked at, yesterday I told you, when I get that, I can go look at the pathways, because my genes next to the region where my signal was is my gene that is what most likely my state is affecting. Okay, so I said, which genes are those? And I say, boom, cell cycle. Of course I expect cell cycle, right? Cells are cycling faster in a tumor and then not, right? Things like that. And then I find many new things that people have not thought about earlier. So I say, boom, LKB1 signaling events or rest of it is also affected by epigenetic gene changes, epigenetic changes, okay, in the tumor state. That's my new finding. And then on the right side, I show you acetylation patterns. You see loss of acetylation on a gene called CDKN1B. CDKN1B is a tumor suppressor gene. So now I have lost the expression of a tumor suppressor gene by changing the epigenetic status of it. How cool. I have found how epigenetic status of something changes the gene expression of a tumor suppressor gene, and that will associate, of course, with my tumor suppression. And I found that happens in a lot of regions, right? So my general conclusion was, in a non-tumorigenic state, I have more acetylation. In tumorigenic state, I had less acetylation. And how do I do acetylations? By histone acetyltransferases, right? So histone acetyltransferase. So I thought, okay, that means when I transition from a non-tumor to a tumor, the activity of a histone deacetylase is increased, right? Did that make sense? Because I'm histone deacetylating, I'm removing acetylation. So I said, can I use an HDAC inhibitor to go on and kill my tumor cell? Okay? All right? So I asked, are tumorigenic cells more responsive to his HDAC inhibitor? And indeed they are. Okay? All right? What does that mean? So my non-tumorigenic cells are all here. Boom. Right? Where? How many of you got that? Do you guys know how to read this graph? Most of you do. Most of you don't. You have grown E. coli bacteria in your life? Okay, I'm sure you have, right? That should be part of your experiment. These are growth curves, right? This is a growth curve of those cells. So now I can inhibit with histone deacetylation and see my green, my blue, and my purple, right? Those are going down. So I treat with the inhibitor, they go on and get killed, but the rest of the graphs don't. What does that mean? That means that my HDAC inhibitor was specifically working in the tumorigenic cells. Did you get that? Okay, that's it. That's the story, right? We're going to end here for now. What did I just tell you? I told you you can start with a biological problem. In this case, a biological problem of non-tumor to tumor and chromatin state differences. And then you can come up with a therapy for my people, right? That's the beauty of science, guys. Okay, you can do this a lot. I just told you some things, but basically I wanted to show you an application of it, because somebody yesterday asked me, can you please do this more? Because then people get it. They get excited as to why the hell should I do ChIP-seq on histone modifications. I don't care, but now you can care because you can build a therapy for people. Okay. And I just showed you a melanoma example. Don't have to worry about it because it's not an Indian problem, right? But you can apply this to anything you want. Okay, all right, good. I'll see you guys in 10 minutes. We're going to start at 12 again, okay?